Okay, today I have a pleasure to introduce Alexandra Weber and her talk she will talk about genomics of sex determination in invasive mussel. Alexandra is evolutionary ecologist, and she's interested in understanding the molecular mechanism generating and maintaining biodiversity in aquatic ecosystem. She used a wide range of omic data from the field and experiment to uncover how organisms adapt to different environmental conditions and also ultimately infer their vulnerability and resilience in the face of the rapid environmental change. She also focused on native and invasive freshwater beaver species in Switzerland to conduct also her current research, but also to have some collaborative project in other aquatic invertebrates from various habitats. So Alexandra, now floor is yours to hear quite a lot about mussels.
Thanks a lot, Elena, for the kind introduction. Thank you to the organizer for the organization of this really interesting conference and for the opportunity to talk about my research. So as mentioned today, I will talk about genomics of sex determinations in 2 invasive mussels. So I will start with a very quick introduction on these 2 mussels, if some people in the audience are a bit unfamiliar with these mussels. So they are freshwater invaders, very successful. They both arise from the Ponto-Caspian region, so that the regions of the Black Sea, and they colonize, say, continental Europe and also North America in the last, let's say, 30 years. If you're interested about these mussels, you have 2 really good reviews that were relatively recently published that talk about these mussels. And they are invasive, so they have very negative impacts on ecosystems. So basically, since they are mussels, they are filter feeders, and they are very efficient at removing phytoplankton and zooplankton from the ecosystem. So they basically drive really big changes in food webs. But they're also infamous because they cost us a lot of money, and this is why they are so famous. Basically, they grow millions of mussels. You can see beds like this. This is really impressive. And what they do, they damage water infrastructure, and eventually they clog pipes, which leads to millions of costs to replace the infrastructure or just develop infrastructure that are mussel-free. And as with many invasive species, it is currently not possible to eradicate these mussels once they have colonized an environment. And then now what has been developed in the last years is genetic biocontrol that could be a solution to eradicate invasive species in general, not just mussels. And what do I mean by genetic biocontrol? It's about disrupting the reproduction of invasive population by releasing genetically engineered individuals in the wild. So if you are interested in the topic, there is this recently reviewed here, it's difficult with this pointer, that talks about the various techniques of genetic biocontrol. And one of these techniques is called sex ratio distortion, and it is exactly what it sounds like. Basically, you would release males, let's say genetically engineered males in the lab, then you put them in the environment, and then this would lead to increase in male frequency, and eventually, when you only have males in the population, then the population crashes. And there is a really nice example of this with these invasive brook trouts. So genetically engineered YY males are produced in the lab where they have actually 2 Y chromosomes. And when you see that when those YY males reproduce with, let's say, XX female, the progeny is 100% male. So that the increase in male frequency is much faster in the environment. And so there have been modeling studies and also pilot studies that show that this is could be a really viable solution to drive populations to an invasive population to extinction, and there are ongoing tests in the fields. But as you can see here, this technique relies on the existence of sex chromosomes, so genetic sex determination. And we touched a bit upon that in a few of our presentations, where we saw a bit of chromosomes, but there are actually a large variety of sex-determining systems in eukaryotes. So the most famous ones, the ones that we all know, is our sex-determining system with sex chromosomes, X and Y, with a male determining locus on the Y chromosomes. But we have also the opposite, that was also mentioned in some other case studies, that there are some ZZ, ZW sex chromosomes where there is a female determining gene on the W chromosomes. Then we have things that can get more complicated with polygenic system with different genes on different chromosomes that together act to have a sex phenotype. And this is also something most people have heard about, is environmental sex determination in some reptiles and turtles, where typically temperature can drive to the development of males and females. And to make things even more complicated, we even have some organisms where we have genetic components and environmental component that drive sex determination. And I think you can get to the point where I'm going, that is, we don't know what sex determination mechanisms are in quagga and zebra mussels. So, oh yeah, and genomics is actually very useful to uncover potential sex determination mechanisms. I think maybe some of you have resequenced populations and suddenly you find weird patterns and you realize, oh, I found the sex chromosome or sex locus. You can do this with a bit more intention. And this is also a really nice review by Beatriz Picoso that was published in 2019. That shows that depending on the differentiation of sex chromosomes or sex locus, let's say, we will see different patterns. So in, let's say here, and where we have just one sex locus that appeared, but then the rest of the chromosome X and Y is still recombining, we will find differentiation on a very, very small region. So these sex, let's say, mechanisms are really, really young. So we have an undifferentiated X and Y. And to detect this, the most common thing to do would be to calculate FST between sexes. Then when there is bit more differentiation between these two, then recombination stops, and then we start seeing that there are differences in male-female heterozygosity, where the heterogametic sex is heterozygous. And then when the chromosomes are very different, when there is a lot of degeneration, we see physical shrinkage of the sex chromosome harboring the sex locus. There we can see differences in male-female coverage. Here. And this is typically in systems where sex chromosomes are very old. And in those cases, we say, let's say when the X and Y are undifferentiated or very young, we say that chromosomes are homomorphic, so they have the same morphology. And when they are differentiated, when they are morphologically differentiated, we say that they are heteromorphic. And what do we know now? Moving on to bivalves. So there are, it's a big, diverse group of mollusks with over 25,000 species. We have been talking about karyotypes yesterday, and so there are a few karyotypes of bivalves. There are 150 that are published, and what we know is that all of the bivalves that have been studied for that have homomorphic chromosomes, including the zebra mussels. You see the karyotype of zebra mussels there. And we didn't have data for quagga mussels yet. So the hypotheses are either we do have sex chromosomes, but they are young, or we can have polygenic sex determination, or we can have even environmental sex determination, or we can have several factors, genetic and environmental factors. What do we know as well from bivalve sex determination from this very recent study that has just been published this year? In this study, the authors looked at an evolutionary sequence of some gene families that are known to be involved in sex determination in vertebrates and also in insects. These are the DMRT families, SOX and FOX families. They have been investigated in 40 bivalve genomes. And there the author found that there are some usual suspects, so some genes that seem to be recurrently involved in sex determination in bivalves. So for male determining genes, these are the DMRT1 genes. So this is a gene that is specific to bivalve. But there is actually another gene, the DMRT1 gene, that is a master male determining gene also in mammals and in fish. And SOX-H are, let's say, usual suspects in male sex determination. And one gene was FOX2, and this seems to be a master female determining gene. So please remember this name, it might come later. Female determining gene. What we also know from that study, and I found that particularly interesting, is that this master male determining gene, DMRT1L, is absent from the zebra and quagga mussel genome, and actually from the whole family. So it is really a true gene loss that happened a long time ago. So if there is male sex determination in these mussels, it should involve another mechanism. Yes. So we started with generating a reference genome for quagga mussels because, well, there was one draft genome, but it had like 80,000 scaffolds. So we decided, some colleagues of mine decided just to do their own new chromosome scale genome. I just go very quickly over it, but we managed to have a decent genome with 16 linkage groups, relatively good BUSCO scores with over 92% completeness, quite a lot of gene models, but this was using transcriptomic data. And some collaborators also generated the karyotype for that study, and they found that they also had 16 chromosomes like zebra mussels. For zebra mussels, this was already done, so we had a chromosome scale level genome and a karyotype that you saw before. So then we first compared the genomes of those two species, and apparently they are very stable because we have the same number of chromosomes. They are all homomorphic, I mean, no differences in size among them, so no obvious sex chromosome. And also similar genome sizes with 1.6 gigabases of genome size estimate. The assembly was a bit bigger for zebra mussels, but this, for quagga mussels, this is the assembly we reach with also over 96 and 98% of the scaffolds that were put together on those 16 linkage groups. So then what we did is a synteny analysis where we do this, whole genome alignments, and we thought, oh, everything will kind of be the same. But actually, no, we found a lot of genomic rearrangements. So some chromosomes were quite, let's say, stable, like this, the red one. But then there was another one, we can't see, but basically coming from the right side, here you have linkage group, that's what, 5. And it has like genes, like homologous genes that were on 10 different chromosomes in quagga mussels. So it means that there are lots of genomic rearrangements happening between those two species, despite the fact that they diverged relatively recently, about 10 million years ago, that they have the same genome size and the same number of chromosomes. Which leads us to think, what are these sex determination mechanisms conserved or are they divergent? So what did we do next? Moving to zebra mussels. We collected males and females from one Swiss lake, and we performed whole genome resequencing. Then we did a classical pipeline of mapping to the reference genome, SNP calling, and we had quite a few SNPs in the end, about 24 million SNPs. And then we identified candidate sex locus by doing first a frequency-based inference, by doing this male-female FST that I showed you before, and also by identifying sex-linked SNPs for both types of systems. So for ZW SNPs, that would be SNPs that are heterozygous in females and homozygous in males. And for XY SNPs, that would be heterozygous in males and homozygous in females. So this was just frequency-based inference, but then we wanted to use this latest model that had been published not so long ago. It's called SDPOP. You have the reference down there if you're interested. And there you can calculate the likelihood of a model following a ZW, versus the null model, which is the model of no sex chromosomes. And then you can compare the likelihood of those two models, and we did that over windows of 10 kilobases. And we did also the same for the XY model versus the null model. Sorry. So moving to the results, this is the results for zebra mussels. We have the linkage groups here on the y-axis, and in black And gray, you have the FST values. We would expect that FST is about 0.5 for sex locus. And on top of that, we plotted the ZW SNPs, so the ones that are heterozygous in females and homozygous in males, because it seems like it's where we could see really clusters and peaks. We did that also for the XY SNPs, but it didn't really look like that at all. It looked a bit cluster all over the place. And then we did the model-based inference here. And here it seems like we have those two linkage group 5, and linkage group 13, that had really signatures of, let's say, ZW sex-determining system. So it seems that in zebra mussels, we do have a polygenic ZW system, because it's on 2 different chromosomes, and we have several genes on those different chromosomes. Now, looking at what those candidate genes are, this is linkage group 5, and you see those 3 really nice peaks. And the first one, so those 3 peaks corresponds to 3 genes, so it's really, really small regions. And this peak was annotated as a Tudor domain containing protein that has been shown to have a role in sex determination in other species. Then the second one is this FOXL2 gene that is a known female sex determination gene, so the one that I was showing you in the introduction. So when we saw that, we're like, okay, this is really strong evidence that the candidate we have is the right one. And then we had a third gene that was known to be regulating transcription, which is what usually these sex-determining genes do. And then moving on to linkage group 13, here it was a bit more messy. It seems like there was especially this first region that has strong differentiation between males and females, but still we had a few clusters of ZW SNPs in there. There, the region was a bit bigger with 21 genes that were annotated in there, and we found 5 long non-coding RNAs. And there are a few studies showing that these long non-coding RNA could have a role in sex determination in other species. We also found a few a bit further down, and here 7 genes with 1 transcription factor. And here in the end, we also had 1 long non-coding RNA, which seems to indicate that we have a ZW polygenic system in zebra mussels. So now what did we find in quagga mussels? So we sampled also 20 males and 20 females from a different lake, because they did not invade yet the lake where we had the zebra mussels, but I hope we managed to protect that lake for a few more years. We did the same pipeline, basically whole genome resequencing, alignment, and SNP calling, the new reference genome that we had. And there we had many more SNPs. We had 42 million SNP, which was very diverse species. And we did the same pipeline, the same analysis with the frequency-based inference and the model-based inference. And here we were really surprised to see that we didn't have evidence for a polygenic ZW system like we saw in zebra mussels, but here we had more evidence for an XY system. It seems a bit messy, but if you look a bit closer, there is like a region here on linkage group 13 that is really a nice FST peak. It has really a nice cluster of XY SNPs, and using the model-based inference, it is also a very, very strong peak for the XY model. Yes. Yes, and then we were wondering, okay, this is on linkage group 13. Maybe it's the same thing as in zebra mussels, but actually, if you remember this synteny plot, linkage group 13 of zebra mussels and linkage group 13 of quagga mussels are really not the same at all. Because here, linkage group 13 of zebra mussel is the green one, and here you can see that the one of quagga mussel is composed of many, many colors, like blue and yellow and red and everything. So it's just by chance that it's number 13. Let's say. Yeah, now looking a bit closer at this region, there the region is much bigger. If you remember in zebra mussels, it was regions of maybe 1 gene, of 10, 15 genes. Here the region seems to be larger, it's about 1 megabase. And there we had 52 genes in there. And that is also what might happen with your annotations when you do annotations of non-model species, that you have a lot of gene models, but you don't know what they do because those genes don't exist in your, they haven't been annotated. So we had a lot of, I have to say, there are a few genes that are not annotated in this region. But what I was most puzzled about is to find FOXL2, if you remember, this gene is a known female sex-determining gene. So when I saw this, I stopped and I checked everything again. I went back to my notebook saying, oh, did I sex them wrong? And then I checked the sex files. So if I didn't, by chance inverted the males for the females, I even did some reference-free analysis with k-mers that I don't have time to show here. But basically, it seems that this is solid. This is true. It's an XY system with the males heterozygous and females homozygous. And the FOXL2 annotation is also correct with the electronic annotation. So what I did next is to look at if this was really the FOXL2 gene in this region. So I took the FOXL2 zebra mussel sequence and I blasted that against the whole genome of quagga mussels. And then I found that the true homologue of FOXL2 was not on this linkage group 13, but it was actually on linkage group 8, so somewhere totally different. So what's actually sitting in the middle of this sex-determining region? This is a new gene that I call FOXY, because I think it's male determining, so let's call it like that. And this is an alignment of the FOXL2 true gene, the zebra, the quagga one. And here in green, it means that the alignment is really good, like 97% identity or even more here as well. And this is why the electronic annotation picked the FOXL2, because this region is actually very, very well conserved. And then in the middle here, this was not conserved at all. And this didn't lead to anything. When I blasted it, it was like something completely new. So this FOXY is very likely a paralog of FOXL2 after gene duplication. And it's just sitting in the middle of the sex-determining region. So what does it do? So then we investigated the DNA binding domain, because this is really like the important part of the gene in a transcription factor, which is called the fork head domain. And what I saw is that it was kind of sitting here in the middle of this weird region that didn't have any homology to anything. And so when I checked if this fork head domain was also in the FOXY gene, actually no, there was the beginning of the fork head domain, and then there was an early stop codon here. So it means that there is this is probably not likely that it's expressing a repressor FOXL2 protein. So what is our current hypothesis at the moment? And I admit it's very speculative because I don't have much data to support this. But we think that this FOXY acts as a negative regulator of FOXL2. And how would it do that? When we have here an XX system, then FOXL2 would lead to the female development. And then when we have an XY with this FOXY gene, that would repress through a non-coding RNA mechanisms, the expression of FOXL2 early in development, which would lead to the development of males. So this is our working hypothesis. Yes, so to sum up, I think I showed you that we have zebra and quagga mussels have very dynamic genomes because they have opposite sex-determining systems. So first polygenic versus unique region, and then ZW to XY. And I think why this is promising is that for quagga mussels, we have a single region. So that could be a good target for genetic biocontrol. For zebra mussels, I think a polygenic system would be too difficult to tweak to have like good GMOs. So I would not go down that route. I would try different things for zebra mussels, but sex ratio distortion might not be the way to go for zebra mussels. Yeah, but before going to genetic biocontrol, we would have to actually validate this candidate locus in quagga mussels using experiments. And to do that, we need to rear these mussels in the lab. And so far, my lab has not managed to do that. They're really difficult, but I will get there eventually. Yes, and anyway, if we want to develop genetically modified organisms, we need to master the life cycle. We need to have these mussels reproduce in the lab. And just a few words about stakeholder engagement. So when I talk about this to cantonal authorities or people who are working in the drinking water companies, they're all very excited to have like a sustainable solution to the quagga problem, because quagga mussel is a big issue in Switzerland. Zebra mussel, not so much. But yeah, still we're not there yet. There are still many barriers to overcome. So first, there are lots of technical aspects to overcome, like rearing them, mastering the genome modifications, and then there are some regulatory aspects, like can we actually release genetically modified organism in the wild? So currently, the legislative landscape in Switzerland does not allow it, but we will see what happens in the future. And there are also big discussions with this gene drive about ethical aspects. How ethical is it to drive a species to extinction? Are we really allowed to do it? Should we do it? Yeah, so there are still a lot of things to develop before we can actually have that working and going. And with this, yeah, there are many people I would like to thank for this project, many of my co-authors, but also many people involved in this project, and I thank you for your attention.
Thank you, Alex. Now we are open for questions. Are there any questions?
Thank you. Really nice talk. Actually, for zebra mussel, I don't know if the genome is still the same, but when I downloaded the genome 2 years ago, and I found a lot of problems with that genomes, like 200,000 annotated genes.
Okay.
In the NCBI notation. So, I mean, Fox, I didn't get if actually you can find the Fox L2 also in zebra mussel, or you cannot find it at all. I mean, maybe it can be something related with the gene annotation, actually.
Yeah, no, actually, I did find it in zebra mussels. That's one of the candidates. But I can check if it's also a different copy of it, and I will check the annotations again. Yeah.
Thank you. Any other?
In your homology plot, it wasn't clear. Are you talking about amino acid sequence, or it's DNA or exons that are missing or not aligning between these two?
No, it was orthologs that were aligned. Yeah.
So the protein sequence.
Because I didn't do those analyses, okay. But no, I think it was amino acid sequence.
Okay.
Yeah.
And then this kind of interference mechanism, I guess you're not suggesting like RNAi, but what, recruitment of other factors, or?
What I'm confident to say at the moment is that it doesn't seem it's protein based. I think it's non-coding, some kind of RNA, non-coding RNA mechanisms. Yeah. But I'm not a molecular cellular biologist, so I don't know much about this. But yeah, that's the hypothesis I have at the moment.
So we have an online question. So do you happen to know the sex of the zebra mussel, quagga mussel that was used for the genome?
Yes, I think they were both males.
Okay, because yeah, they were saying this. If it was a female, you would like the ERGA gene. So, and another one.
No, but this gene absence, that's, yeah, you mean that. Yeah, but it was done in a phylogenetic context, and then you could see that almost all of the species that were in this clade did not have this DRMT1 gene. So I think this is a gene loss that happened much earlier in the past.
In a polygenic sex determination system, so I guess that how well GMO-based control works depends on the architecture of the sex determining the lossy. So can you check with your data whether they are dominant?
Whether what is dominant?
The GMO-based control works.
Yeah. So in the GMO, the genetically modified organisms?
Yes, if the sex determining architecture is...
I think in GMO, it's never polygenic. I think when they do it, it's a classic XY system with a strong degenerated sex chromosomes. Because actually true examples of polygenic sex determination are actually fairly rare. So there are a few in fish that I know of. Yes, I think all of these genetic biocontrol with sex ratio distortion that has been done is just on classic sex chromosomes.
Okay, thank you. There's a last question.
Given Switzerland's current thinking on GMOs, are you going to just continue this conversation for the foreseeable 100 years? Or what is your confidence of actually beginning to change perception especially in a situation like Switzerland?
Well, maybe that's because I'm an eternal optimist, but I think anything is possible. Yeah, no, and I mean, in my research institute, we are fairly close to policy and practice. So I think it is possible to have at least this conversation going. And this quagga mussel is such a big problem that it might be, the exception to the rule. Nobody wants this genetically modified maize in the plate, but when it's about saving millions and billions, maybe they will change their mind. Yes.
Money talks.
Yeah. No, it's sad, but that's how it is. Yeah.
Okay, thank you again for a nice talk, and we can move on.